So now that we've covered what a virus is, its structure, and everything like that, let's talk about how it actually reproduces. So this is the confusing part about viruses. So let's start. So how viruses work is they are made of protein and DNA or RNA, and what happens is the capsid goes and it attaches to the host cell. And so here we have our virus. It actually comes and attaches to the host cell, and what it does is it injects its genetic information, whether that be DNA or RNA, into the host cell. And it's very specific binding. So the virus is going to come along and look for specific receptors on the cell's membrane to attach to. It can't infect a host cell that it's unable to recognize, and so a lot of antiviral medicines work by blocking this binding. So if you can stop the virus from actually binding to the host cell, then it's not going to be able to do anything. And this is why it's so specific that a virus only really kind of works on one host cell. And so then it injects its nucleic acid, whether it's DNA or RNA, into the cell, and then the nucleic acid that it's injecting is coding for the proteins of the capsid. So a virus is so simple. It's capsid and RNA, or DNA, and that's really all. So all it needs to do is get its DNA replicated by using the host cell's machinery, and get its protein replicated by using the host cell's machinery. So there's a couple of different kinds of viruses we're going to talk about. The first one are those that infect bacteria, and these are called bacteriophages. That's pretty easy to remember. A bacteriophage has two different life cycles, the lytic cycle and the lysogenic cycle. And so the lytic cycle is the dangerous one. The lytic cycle is when it's actually using the host cell's machinery to make more of itself. And so the really, really virulent viruses, the ones that are really dangerous and really contagious, are those who enter right into the lytic cycle when they enter a host cell. Now, there's this other cycle called the lysogenic cycle, and the lysogenic cycle is when the virus simply integrates its DNA into the host cell's genome, or RNA into the host cell's genome, and just hangs out and isn't really doing very much. Its DNA is part of the host cell, but nothing's really happening with it. So here's the image from your textbook. The lytic cycle has 5 stages. The first one is attachment, when the virus actually comes along, finds the proteins on the host cell's cell membrane and attaches to the receptor. Then the second step is penetration, where the viral DNA enters the host cell. So this is where it's actually injecting its DNA or its RNA into the host cell. Then there's biosynthesis, where it's using the cell's machinery, so proteins, enzymes, the energy from there, everything. It kind of turns off all the cell's normal functions and just turns on virus making. Basically, the cell becomes a virus making factory. As you can see in here, now we have all of these images of DNA, or of the capsids and the virus DNA. And then maturation is when they're actually all combined together. So they're putting together the DNA and release is when the cell is actually going to lyse open and release all the viruses into the surrounding extracellular solution, and then it's going to go and infect all the other cells. So that's the lytic cycle. There's also the lysogenic cycle, like I said. So the lysogenic cycle follows the same 2 first steps, which is attachment. Obviously, we need to find the host cell, and penetration. If we don't introduce our DNA, nothing's going to happen. And then if it enters the lysogenic cycle, what happens is the viral DNA is integrated into the bacterial DNA. So basically, the viral DNA that they injected, the bacteria just takes it up into its genome and now becomes part of it. So it's just another gene that is part of the genome. So if the bacteria replicates, then every cell now has this as part of its DNA, which is also pretty dangerous if you think about it, because now any of those cells at any time has the opportunity to start making the virus. So the viruses that enter the lysogenic cycle first can be switched into the lytic cycle by a lot of different things, like ultraviolet radiation, or anything like that can just cause it to suddenly start going into the lytic cycle. Now, when the cells are stuck in the lysogenic cycle, this is called a prophage, because it has the virus as part of it, but it's not really doing anything. And this is what causes, so you guys know strep throat. If you have strep throat, you are infected with bacteria that causes strep throat virus. If that bacteria, the strep throat bacteria, is infected with a prophage, then if you get strep throat, you'll have scarlet fever. So it's much worse because you're infected with the bacteria and with the virus at the same time. So that's the difference between the lytic and the lysogenic cycle. and there are the steps. So attachment, penetration, biosynthesis, maturation, and release are the steps of the lytic cycle, and then attachment, penetration, and integration are the steps of the lysogenic cycle, the latency phase. So that's the bacterioviruses, the bacteriophages. Then animal viruses are a little bit different because obviously our cells are different than bacteria, and so the methods of introducing the genetic material are a little bit different. So the virus doesn't just attach and inject its DNA because we have a lot of other things happening. So it can do that, or it can enter by endocytosis. So it attaches to our cell membrane and we take it inside. And then what's going to happen is the virus becomes uncoated. So the capsid kind of falls apart and the DNA is just free in there. And then the method of viral release is again a little bit different. So sometimes they can be released by budding, like a big chunk of them will just bud off, or if there's enough of them made inside of the animal cell, it'll just lyse open and release all of the viruses to the surrounding areas. Now, the one main kind, or not main kind, but one of unique kinds of viruses to animal cells are called retroviruses. So retrovirus is an animal virus with an RNA genome. So this virus has RNA, not DNA, that is converted to DNA within the host cell by reverse transcriptase. So retro kind of means backwards. So the virus has RNA inside of it, and it injects its RNA into the host cell. Now, that host cell, we're eukaryotic cells, our genomes are made of DNA, so it doesn't do us very much good to have just extra RNA, right? We could make the proteins, but then we're never going to integrate them into our genome or anything like that. So what happens is reverse transcriptase comes along, and we have our RNA molecule, and remember reverse transcriptase turns RNA into DNA. So we use reverse transcriptase to get our single-stranded RNA into a single-stranded DNA, our complementary DNA, cDNA, and then that's used, made a double strand, and it's integrated into our genome. So now it's part of the animal cell's genome. The biggest example of this that you would know would be HIV. HIV is a retrovirus. So this can lay dormant inside of our cells for years and years and years. I think your textbook says 9 to 11 years without treatment is the normal time for HIV, but it can at any time become active. So the way that we stop these is by using antiretroviral drugs. So it's a virus, so it can't be cured, but we can stop the retrovirus from happening. We can stop the RNA from being changed into DNA. So this is an image right here of the way that HIV works. So it comes along right here, it attaches via a receptor, and then it is, by endocytosis, is brought inside the cell. Then the capsid is gone. Look, there's no more capsid, it dissolves, and we use reverse transcriptase to take our viral RNA, make it into cDNA, and then it's integrated into our host cell's genome. Right now it's a provirus, just like it was a prophage in the other one, now it's a provirus. It's inside our genome, it's just hanging out. Then can be made into mRNA again at any time by transcription. Then out here it's translated, and now we have all of our proteins that we need. Then it can be put back together through maturation and release. So that's how it works inside, a retrovirus works inside of a human cell. So over here. So this some of the most dangerous viruses are those that are deemed emerging viruses. So they kind of just come up from somewhere and become very widespread and prolific. Some examples of these are AIDS, West Nile virus, SARS, bird flu. And the reason for these is because reasons for sudden emergence can be a couple things. Maybe they're transported from one part of the world to another, and that part of the world is not prepared for it, or it can jump from species to species. One of the biggest ones that you probably know are swine flu. There was a big outbreak, obviously, of the swine flu in 2009, but this is, it's just a type of human influenza, which is actually part of the bird flu. And what happened, and there was a horrible outbreak of this in 1918, pandemic killed 50 million people, which was a third of the world's population at the time. And then there was another outbreak in the 1970s, where a lot of people were killed. And the reason that they think that when it broke out in 2009, so many young people were affected was because all of the older people who were alive during the 1970s or shortly after epidemic of the swine flu, they were already had some predisposition to survive it better. So that was the H1N1 swine flu is another example of an emerging virus. So lastly, your textbook talks about something called viroids and prions. And viroids and prions are very similar to viruses, but they're not exactly the same thing. So one of them is the viroid itself is a naked strand of RNA without a capsid. So it doesn't have a protein coat, but it does the same thing. It can't replicate itself, but it enters a host cell's genome and gets it over and over again. And these are very prevalent in plants. A lot of plant diseases come from viroids. Then there's also prions, which cause something called, or they're defined as proteins that normally exist in an animal but have a different conformation. So there's just something a little bit different than them, and what they do is they go around to other proteins and they interact with them and they make them to change their structure so that they're no longer useful. This usually just happens by changing one chemical bond and suddenly that protein is no longer useful. There is no cure for these and they're very dangerous. So one of them is they're called transmissible spongiform encephalopathies, TSEs. One of these is mad cow disease. I'm sure you guys have all heard of mad cow disease. It's very contagious and very dangerous. And so that's viroids and prions are kind of the opposite of each other, like half of a virus. Viroids are naked RNA and prions are proteins that do the same thing. They're not whole viruses, but they act in a similar manner.